So my talk is going to be on disease control and keeping the plants healthy, and working with Dave Schwartz on his research farm, which is a home farm down by Guthrie Center, where he has reached corn that hit 411 in spots and 370 in the whole field. We're keeping the plants dark green until 20th, 22nd of October. Okay. Yeah, keeping the plants healthy up till a hard freeze is the way to maximize yield, both corn and beans. So I'll try to go as fast as I can through this, a lot of information. So topics we'll cover: major diseases, exposed resistance problems that exist right now, and looking into the future. So the main thing right now, and the thing that Don Huber changed my mind on the most, is I was educated. My degree was in plant path, pest management, agronomy at Iowa State. And what they do a good job of teaching you is how to identify a fungus using a microscope and guided books, pictorials. And the next step they jump to is having you decide what fungicide you're going to apply. Now, in my mind, what the next question that makes sense be, can you figure out, or what is causing the plant to not stay healthy and have a fully functioning immune system? So they're missing that step, and I think that's what would benefit farmers the most, because if you hit it with a fungicide right away, it may cost a lot more. You're doing damage to the soil. I showed our test results to Jerry Hatfield, head of the soil tilth lab at Iowa State, and first thing you said, you got to show this to more people, because if they spray tar spot 2 or 3 times with a chloride or fluoride based fungicide, they're going to damage the health of the soil a lot more. We've all seen the triangle, which they say if you have a host plant, a susceptible one, the right environment, and the disease inoculum, you're going to get the disease if it all transpires. Now, when a person hires an agronomist, I think one thing that is necessary is to be able to identify the role of every nutrient that takes place in the plant, and make sure you've got a sufficient level in there, and confirm it with either tissue test or sap analysis. Probably the best guidebook I've seen, or the article that talks about the role of each of the minerals, is by an Australian guy by the name of Graham Sate. And if you want, I was down in Amarillo last week, and I put a couple flash drives together for the people that attended, and I included this whole thing. About an 18-page article, easy reading, interesting reading, and it spells the role of each of them: zinc, copper, silicon, what have you. And Dave Sasseville, who was a micronutrient expert at the University of Missouri for the whole Midwest, put things in perspective and led some logic to what has transpired. And a lot of the heavy-duty fertility research was done by the old masters, and they did this work starting in about 55, and they finished their 10th year in 1965. And what they said at the meeting with the press there is we have enough micronutrients in the soil for about 40 or 50 years. You average them 45 years after that date, something is going to happen. And what the press heard and what they wrote about is you have enough micronutrients in the soil forever. You don't have to worry about them. And people ask me if I'd like to have a tissue test on the fields I'm working with. To me, if I'm flying in a jet airplane at 450 miles an hour, 30,000 feet up, I want the pilots to look at the gauges once or twice at least. And taking a tissue test to me is like looking at the gauge. But anyhow, the major disease problems that begin to show up Midwest-wide, if you take 1965 and add 45 years to it, that says the major problems would appear in 2010. That's exactly when we had major disease problems, both corn and soybeans, from Omaha, Nebraska to Beltsville, Maryland. So Don Huber, a lot of people have mentioned him. I've been able to be blessed to spend a lot of last 14 years, quite a bit of time. And his background is, well, Mark had him for class, a few other people have, is he was at Purdue University, Professor Emeritus for 35 years. He also had a concurrent assignment as a medical expert for U.S. military. He was head of the Threats Pathogen Committee and served on the nuclear disarmament Russian inspection team. But he wrote a book co-authored a book called Mineral Nutrition and Plant Disease. It's very good, 21-chapter book, and it talks what happens when you've got a deficiency or an excess in different minerals. So if you go through one chapter in a week, you've read the whole book in about 4 or 5 months, and you understand things a lot more. Now, one book that's worth looking at is called Tuning into Nature by Phil Callahan. And Phil started out by being in the Air Force. I don't know if it was in the US or England, but he had to conceptualize, design, and build insect or radar antenna that would pick up enemy German bombers getting ready to drop bombs on the cities. And after he got done, he went and got up to his PhD in entomology, and he was looking at the radar or the antenna of the insects, and he said, these look an awful lot like the radar I've been designing for airplanes and to identify airplanes. And he asked the question, is it possible that the insects are navigating to find their mate, find their food, and finding their way around using radar? All the other entomologists said he was crazy, but the one group that paid for his last 35 years worth of research was the U.S. Air Force. So all the technology for new radar and stealth technology came from an entomologist down there. His prize pupil is Tom Dykstra, who runs an insect research lab down at Gainesville, Florida. He's in our think tank. I know him quite well. Him and I and Steph Seneff took a high school kid science fair project, and we guided him. It was on earthworms, and took him to second place in the U.S. for that category. It was kind of fun. Now, typically we look at number of pounds or pounds of nutrients that are pulled in by different size crops. And what we can use is either tissue or sap testing. Sap is probably the best, but you have tighter regiment you have to follow. You have to get the leaves to the shipping place, FedEx, by the end of the day, keep the labs cool. And what they typically do is they track the mineral levels in the new leaves versus the old leaves, and you see which one is going up, which one is going down, and you can basically track what is being utilized by the plant. Tissue testing, Ward Lab in Kearney is where I send my samples, runs about $30, and I have Molly run on every soybean and corn leaf because it is deficient 95% of the time. SAP, there's about 4 labs working in the U.S. Mark, there's one in Texas, one in Michigan, one in Washington State. Is there a 4th one right now?
Not to my knowledge.
Okay. Mark is doing a lot of SAP, so when he gets down into Texas, they got high aluminum, high chloride levels. SAP is probably the way to go, but it's tougher to look at it and totally figure out what is going on. Question comes up. There's a lot of GM crop, or a lot of acres of GM crops out there, both corn and soybeans. And a good question to ask is, when they insert a trait, does it affect the mineral levels in the plant? If you're going to do that sort of research, you had to get written permission from the seed companies to do it. Well, they weren't going to give permission, but it was done by 2 individuals. One was Luis Zolvioli, who was a young postdoc student under Bob Kramer at Nebraska, at University of Missouri. I went down to meet him because Bob said you need to meet him because he got more work done in a year than most other grad students got done in 3 years. And when I go down to Brazil, I usually look him up. But he found out that when you insert the Roundup gene, the zinc level decreases by 47%. and the manganese level goes down 17%. Zinc is highly important in root health, disease protection, and also drought tolerance. So when you have a Roundup gene, you've got to respond with foliar zinc more often, and you need to watch it if you're going to have plants that take the dry weather we've been having lately. Manganese allows the plant to, first of all, wall off an infection peg from a fungal spore if it germinates and is trying to get into the plant. And then it also allows the plant to split water into hydrogen and oxygen and use it to make sugar with. The other guy that did work was from Turkey, and I guess they figured they'd be out of the country before the compost hit the fan, and they couldn't track them and they were safe. Katmak was actually working as a grad student under Purdue, or under Huber at Purdue. When we do our summer lab analysis, we use what's called an X-ray diffraction scanner made by Bruker. It's a $35,000 instrument that was repurposed by Joe Clapperton for use in ag. Looks like a giant squirt gun. It's so complicated to run that Lauren Steinlage's 9-year-old daughter got real good at running it when she was in the state a couple years ago. But what you can do with it is lay any grain sample, any plant sample, any food sample on a tray, press the trigger, and in the old days it took 90 seconds to get the full reading of everything atomic weight 12 and heavier. It doesn't pick up boron though, or nitrogen. Now, Jill did some updating on the software. It now happens in 30 seconds. So I think this will totally recognize fertilizer research, because you don't have to pull leaves and send them off to a lab and wait 10 days or even a week to get results back. You can walk through the field and test the leaves and have the results in 30 seconds. We met with her and I asked, how long will it be before you've got a high clearance sprayer that has direct injection, maybe 5 or 6 different tanks and minerals, and you've got a boom in the back that is applying exactly what the scanner parked on the front of the machine is telling you it needs. Her response, we've already got it designed and patented. Now I think we're going to see drones set up to apply micronutrients in the future also. Now yesterday we had Liz Haney talk for a little bit. She was at Mitchell's Field Big Soil Health Day over in eastern Iowa. Monday and Tuesday she stopped in to stay a little bit and explain what regenerative ag is. And Rick Haney, her husband, started out as a feedlot cowboy, an electrician, a furniture upholsterer, and after so many years I think got tired of getting dirty and bruising his hands and cutting his hands all the time, so he went and got his way up to his PhD, and he finally developed a test that would let a person take a soil sample and analyze it for analyze it besides mineral content, figure out what microbes were in it, how active the soil was, and what we like to see is at least double digits on the soil reading, and the highest I've ever seen was a 29. Guy up in Minnesota hit a 25, and my question for the guy that hit a 25, how long ago was the last livestock operation there, and he was still putting manure on from a hog setup. The 29 was with the guy at southern Minnesota, and he had had a dairy there 30 years before. So the effect of higher biology was carrying through that long. Now, Agronomy RX is working with Dan Coffin, who was a previous formulator. We like what they've got. We're using them for a mineral supply. And Vachi, who you met, is working with Andy, and they've got the fermentation tanks, and we're getting the microbes and the minerals from them that we use in foliar feeding. So we like what we're seeing, and this is what we had last year. And say, for example, when we do a tissue test, what we saw last year, fell over in the second table there, put microbial mix on his soil for the first time, and we can typically see the soil health reading jumped from maybe a low, say 1 to 5, up about 7 or 8 points with just one application, 1 year. And I talked to Jerry Hatfield, and I told him that his axiom saying there's no such thing as bug in a jug, I think was outdated because of what I've seen with putting on, say, a Biodyne mix or a Agronomy RX mix. We've seen that much of a change. Evan over there, his tissue test readings in Wisconsin were on the low or very low side. After one application, most of his tissue test levels were in the medium category or medium high. They jumped that much just because the microbes were finally there and able to release what was in the soil. That's one of the key components in the regenerative ag moment, because say we had a Russian fellow that has visited this spring, and he writes for Russia Today, and the question that he had when he came out here in May is, how did your farmers you work with compensate for the fact that fertilizer supplies from Russia and Ukraine and those bloc countries were not as plentiful as they were in previous years? And they typically responded, we were okay in 2022 because the supplies were already there. It's 2023 we're worried about. And he asked me what I thought, and I said, my message for Putin is that we're going to get by, and if the regenerative ag movement continues to be highly successful, and guys find out that they can make the minerals in the ground more available, farmers may find out they don't have to buy as much in the future, and Putin will have shot himself in the foot as a result and wish he hadn't done that. I thought that was probably the right response. Okay, biologicals are more mainstream right now. We're using them, we're depending on them. Still, I think the one fault is when they are tested initially and they're sold, they never give the instructions that a farmer needs to evaluate each field as far as what the previous herbicide history was, and also look and see what the Hades score was originally. So say they were doing some in vita testing. I talked to the head agronomist and I asked, are you tracking those two things when you put out a plot? He says, no, why should I? I said, everything is predicated on surviving and living long enough to do the job. So pay attention to, say if you got a lot of Roundup residue from the past, say in Australia, where they got real bad soils, real dry, they got certain types of clay, but they had an area that they tested where they had put on 2 pounds, 2 quarts per year, which was 2 pounds, each year for the last 22 years, 44 pounds total, and 41 pounds were still sitting there. So I would not want to sell a biological to that fellow. That's why having Martha's stuff available with the sauerkraut juice is something we finally have. Now, why am I somewhat paying attention to having such a heavy reliance on hard fungicides? A couple reasons. Mike and I were selected to be on the USDA Soybean Rest Task Force, so I've spent about 300 days in South America working with top pathologists. And I've seen it down there where one field of soybeans in a town called Primavera del Este was getting sprayed 11 to 13 times one season. If they didn't spray it, it was going to die. Now in that area, the strobes and triazoles are completely ineffective. And they came, in this country, they introduced what's called the SDHI, or the carboxamide family, to come with the 3 ways, Trivapro, Veltima, all 3 ways. We were there in February of 2017, and I asked their epidemiologist in Brapa. We were looking at plants that had been sprayed 3 or 4 times with a 3-way mix, and they were heavily infected with soybean rust. And I asked Claudia, I said, it looks like you have a little problem here with resistance. She shook her head yes. And when I asked her what's plan B, she goes, I don't know. So currently all of the hard fungicides sold in southern Brazil by Bayer, BASF, and DuPont are mixed with Streit products, which are amino acids, minerals in a phosphite chelating mix, and we'll get more on that lately. Now, if you're old enough, you probably played around with the fungicide family or used one called the dithiocarbamate family. Names for them were Manab, Mancozeb, Zynab, and I sprayed them, I used them, I had to wear a mask, I had to wear a suit, and I had to wear a respirator. They cause cancer and they cause glandular problems, and as a result, most of them were pulled from the marketplace. The active ingredients in those, though, were copper, magnesium, boron, zinc, and iron. Those minerals have fungicidal properties. So, got a picture here. First of all, in the top left, we were down in a field in Brazil. On the right, top right, soybeans that were infected by soybean rust. And what the disease is, it was number one on the threats pathogen watch list. But when you start to see some yellowing, the field is going to die. You can't do a thing about it. As a result of the trip, I got invited over to Bayer headquarters, spent days with their top scientist, was talking to Albert Witzenberger, fungicide product manager, and he asked what I thought of their commercials. I said they don't resonate with American farmers, so I said, he asked, what should we do? I said, get a guy that looks like John Wayne or Liam Nielsen doing a fist bump or something. I should have trademarked because that was their head commercial there for a few years. But I did partake in what was called an aerobiology study that was run by Scott Izard out of Penn State, Joe Russo, who ran an aerobiology company. And we set up spore traps clear across the country. I was in charge of the ones in the northern part of the country, and what we took was a large plastic funnel. They put like an old milk filter in the bottom, and it caught the spores that were coming out of dropping from the atmosphere in the rain, and we were catching live spores all the way to the Canadian border. Now, they were alive, but they had been sterilized once they got up to 1,000 feet by the sunlight. Now, talking to Antonio Ivankovich, the head pathologist with the Argentine USDA, he said they're finding out the spores are gradually forming pigment. So we cannot say we're home free on this one. That's why we need to keep developing things. Got a picture of field in the lower right that was around a town called Novo Mutum, somewhat north of Cuiabá, south of Sorriso, but that field made 0 bushel per acre. Another thing that doesn't show up in the farm magazines is the medical aspect of the current fungicides. At University of North Carolina Medical School, they exposed mammalian nerve tissue to headline fungicide. It was causing nerve damage. Also, how many people have heard of Candida auris? People know what Candida is. This is a species that was running wild in doctors' offices, hospitals, and clinics in New York, New Jersey, and Chicago at that time. And I've got a sister who teaches infectious diseases at University of Iowa Medical School. I knew about it 9 months before they did, or she did, but it was causing 61% mortality. And we worked with an Oxford-trained medical forensic pathologist for a number of years. I told him we were using triazoles for crops, and he just looked at me horror-filled. But he would never explain why. And I found out, though, most of the medicines used to treat fungal infections in people are triazoles. So there was a talk at the American Phytopath meeting a little bit over a year ago by 2 professors at University of Georgia. And what they're saying is the prophylactic application of fungicides is causing resistant strains to develop that will eventually go after people. So I see probably a meeting in the future where you've got on one side, you got CDC and AMA versus USDA and EPA debating whether products should have more value as an animal medicine or plant medicine or a human medicine. We all know who's going to win that argument, probably rightfully so. So the bottom line in there is it says a desperate need for new environmentally friendly fungicides. I wrote them a 3-page letter. They're saying we need a lot of funding, a lot of work. I said, the work's already done. We know exactly what the answer is, and that's good mineral nutrition in the plants. One thing I learned a lot from, this is a research article on enhanced instruments, sensitivity and selectivity for aminopeptase profile. What the heck is that? Well, I got involved, Don and I were down in Brazil, courtesy of Streit, and this was February of 2017, and we went to their greenhouse with their pathologists and physiologists. They're an innovative company. They work on plant nutrition, plant health, and how this whole thing works is when you have a phosphate or phosphate fertilizer, it's a PO4, and that means there's 1 phosphorus and there's 5 oxygens on a single molecule. If you take away one of the oxygens, then it's a PO3. It's no longer a phosphate fertilizer. It's a systemic product that will go into a plant and then move up, so it's systemic. And benefits, it's soluble, moves in the plant, moves around the plant, lasts a long time. You don't have to worry about how many gallons per acre you're putting on. And then there's amino acids. There's 304 of them. Plants can absorb 11, and the plant needs 20 total. Each of the amino acids has a specific purpose. We use a lot of glycine for flowering and fruiting, tryptophan and glutamic for germination, but they know exactly what mineral amino acid does what. I can send this out, but what you do is you get systemic protection. They move around in the plant. You don't have to worry about volume and coverage. What we saw in the lab is they had a big black slate tabletop completely covered with petri dishes, and if you reach down by your left pocket and extend it clear as far as you go with your left hand, that whole left side was one fungus after another that affects crops. Pythium, Phytophthora, Rhizoctonia, and what have you. And then as they started to move to the left, they put in a phosphite, and the disease circle in the center got very, very small. And as they continued to put in individual minerals and then combinations of those minerals, copper, magnesium, boron, zinc, and iron, the disease circle got smaller and smaller. And then they started to add amino acids, and when they got the right combination, the disease disappeared entirely because it was screwing up the nitrogen metabolism of the plant. So we got outside, and I told Don, hey, I've seen a lot of demos, but never one quite like this. What am I looking at? And his Rolodex was running. He said, well, back in the era where we had Cold War with Russia and North Korea, which would have been the late 70s, early 80s, we were worried that they were going to drop pathogens on our crops to deplete our food supplies. That's why herbicides were developed, 2,4-D during World War II. And he knew we needed a quick, easy, effective way to identify each of these. And then what they did, they built a pulse laser. They did a complete DNA scan, complete amino acid profile. When they hit the right combination, they wrote the recipe down. I asked what happened to the recipe cards. He said, they're sitting in a cardboard box up in my garage. Do you want them? I said, they're national treasure. We can't throw them away. But anyhow, that's what Streit is using, and that's why their products are good. So we took all that knowledge home and used it when we started to see different diseases here. Now, what we tend to do is when we're walking through a field and we see a plant that has a lot of very obvious streaks in it, then we know usually zinc, manganese, our deficiencies are showing up. And talking to the guys at Midwest Lab, moly is short 95% of the time, boron 90, manganese 85, zinc 75. And if you don't basically eliminate those deficiencies by putting on micronutrients, the plants are going to get sick and the grain is going to be deficient, which will turn into a deficiency in the animals that eat that feed, and then probably deficiency in the people that eat that animal. Yes?
Back to your phosphite amino acid, does certain minerals work better with a certain chelation?
Yes. Yeah, we worked with a German company and we could not get their glycate chelated manganese in solution because we were using indicate 5 to chelate it. And talking to the head chemist with a company from Utah, he said anyone with a lot of experience knows you have to use methionine to chelate manganese with. So we're going to see more and more of these. The beauty of it is you can get by with ounces per acre, dramatic effect, and I'll show you some stuff that's pretty much unbelievable in results. But tissue tests, we like taking them because soil tests tell us what's in the ground, tissue tests tell us what is getting into the plant. One disease that is still affecting us, and between 2010 and 2017, I'd say 70 to 80% of the corn south of Highway 20 was dying 4 to 6 weeks early and down quite a bit in yield. And to this date, no pathologist from the university or extension has said what was causing it to die early. Was it committing suicide? Was it standing on the ledge on a high skyscraper jumping off? We worked on it quite a bit because I had a background that dated back to the first BT experiments. I had some insight on it. The disease is spreading field by field, state by state, through the seed supply. And I've got pictures taken over Fort Dodge, Iowa from Dave Olson's airplane, which he sat over there a little bit earlier. If you can look in the field, our picture, you can see these circular spots that are spreading downwind from a certain plant in about a 50 to 80 foot circle, and those are dead plants. So the universities say only 3% of the seed remains infected in the spring. Well, if you take 34,000 seeds per acre times 3%, that tells you you're going to have, what, 960 dead spots out there. And if they reach 50 to 80 foot across, that's a high percentage of the 8 acre field that is going to be dead early. Is this going to stop? No, this is a seed field that a friend of mine down in southeast Iowa took a picture of this fall. That field has not been sprayed with a desiccant. That is very heavily diseased. If they put the seed in the bag, is it going to disappear or stop? My wife and I were down in St. Louis a couple years ago. I love the first sweet corn of the season. We went into a Schnucks supermarket and they had some fresh sweet corn that was growing from seed down in Florida. We picked through the ears and we start finding a lot of the ears had Gauss's wilt lesions on them. So the universities don't want to talk about this because it's against state seed law to move it across borders and federal to move it across federal borders. Anyhow, the symptoms are different than is listed in the paper because we're looking at a gray, grayish-colored, caramel-colored lesion that marches from the ground up, starting at V10 to V12 growth stage. So it comes in there early. What we see are great big thumb-sized black prints on the stems or on the stalks. Once you start to put on the ears, what it did for standability, it totally destroyed it. And how it does it, it produces an enzyme called amylase, which is used in an ethanol plant to dissolve more of the fiber and cause a greater ethanol yield per bushel of corn put into it. And this was by Marshalltown, Iowa, with guys that typically raised 240 bushel of perfect standing corn. I was riding with them in the combine. They have a lot of point rows and terraces, but about half the time on the last 500 acres, they could not tell if they were going with the row or against the row. That was hell for them to get that done. So we ended up coming across, through Don's help, a biochemist up in Logan, Utah, who had developed a product against Clavibacter bacteria, which caused the bacterial leaf ring spot in potatoes. If one potato in a whole storehouse full of potatoes was infected, within 6 weeks, all the potatoes will be rotted inside. Back in 2014, we tested 30 acres worth, got sent to me. A couple neighbors had late planted corn and high clearance sprayers. We saw enough benefit from it. We thought, let's try it again the next year. We did. They shipped us 2 totes of product. We had it applied on 2 guys' fields. And lo and behold, those fields stayed dark green and healthy an additional 4 to 6 weeks. And the stuff yielded 90 bushel an acre more. We knew we had something good. That's what 340 and 370 bushel corn looks like. And every stalk standing perfect. We also correlated B14 genetics were more susceptible, especially the smart stacks. We also did some heavy duty sleuthing, and we took Gauss's killed corn tissue and sun death killed soybean tissue into industrial scanning electron microscope lab and took pictures at magnifications from 14,000 to 200,000 times. And we saw a lot of crystals in there. And we're working with a vet that did the autopsy on the secretary at the racehorse. But anyhow, those crystals belong to a semi-metallic mineral class. And we know they couldn't cause problems, but they were in the same class for what caused mad cow disease. So if we look at the fungal disease problem in the Midwest, most of the U.S., we've got gray leaf spot, anthracnose, and so on, but they're all endemic. We know what they look like, and we can diagnose them, but do we want to keep using the fungicides we've got, or do we switch gears a little bit? First of all, what are fungi? Fungi are plants that they did not receive any chlorophyll. Therefore, they can't form their own sugar, and they have to seal them from plants that have their own chlorophyll that can make sugar. So they become parasites. When you take a mycology class, you look through a microscope about 3 hours a day, 4 days a week. You learn all of them, very intricate. In the bottom right, there's some beaker dishes, and when they start to put Roundup on the soil, you get a lot more fusarium growth on the roots. Bob Kramer and his work at Missouri basically verified that if you put a lot of glyphosate on, you kill off the Pseudomonas, that is the microbe that turns P2 phosphorus into P2 phosphorus, making it available. It also puts off 4 compounds that are fatal to the Fusarium fungus. Mycorrhiza fungi, what are those? Besides good bad fungi, there's good ones. One we like to put on the seed that I tell farmers use if you're looking towards or expecting a dry year, because it grows a larger root system that expand more territory, seeking moisture, seeking minerals to help produce higher yields. And they're very complex, probably one of the neatest things I've run across. And why they go after plants is they don't have those 5 basic minerals that they use to stay healthy. But we've seen a lot more corn diseases in the last 10 years because usually micronutrient shortages cause more to happen. In the case of gray leaf spot, say Pioneer 3394 was the king hybrid for quite a few years in the U.S., but once people went to more conservation till, left a lot of stalks on the ground, we had a higher level of inoculum every year, and it happened to be quite susceptible to gray leaf spot, so that pretty much killed that hybrid. Northern corn leaf blight is when we see a cigar-shaped lesion on the leaves, and again, some of the B37 hybrids quite susceptible. Eye spot, more of a disease that occurs in Wisconsin where you've got a lot of dew, a lot of cool temperatures. And normally not a major problem, but we have seen them. One year got quite bad. Physoderma stalk rot is on the increase. At first it was just a few stalks on the leaf axil. Now we're seeing quite a few on the stalk, and the stalks get quite brittle. I would expect it probably to keep increasing in severity a little bit. One worth noting, you see that green leaf with a lot of red dots? That's southern rust. And it flies in typically mid-August, late August, southern part of Iowa, and it can get quite a ways north. Usually comes in out of Oklahoma, Missouri, Kansas, Nebraska. And once it comes in, if the weather is moist, you can have a field dead in 7 to 10 days if you don't treat it. Now, Mark and I were down south, and they had different crops that they work with: melons, potatoes, tubers, anthracnose, and they have to be more careful, more specific, because they're human consumption products. In 2020, a German company moved a product in called phytobiotics, and they were using, again, chelation or complexing, using those same 4 minerals. This is what those leaves look like. We showed that already. And these pictures were taken October, like around the 25th. And I told the 2 brothers, you've got to do something with that southern rust, because the whole field is just in the late milk, early dough stage. And if you do nothing, you've got 100% yield loss, because it was 2,000 acres of late planted corn. I said, I just looked, but they said, we've asked, and there's nothing we can do for that. I said, well, I just looked at 1,400 acres in northwest Iowa. That looked awfully good. And we've had that on the plants for about 7 days, so I think it's worth a try. They applied it. I had the farmer watching quite closely. We put on 2 ounces of copper with an airplane and 2 ounces of water, or 2 gallons of water. The lesions began to disappear in 30 hours, and a week later, 95% of leaves were completely green, completely healed up. I've never seen that before, but having curative activity and total systemic activity takes away all the questions about how much water do I need, can I be early, can I be late. They also have these leaves stay that green for another 40 to 50 days. And take another shot right there. Almost unbelievable from within a week. So that's a benefit. That's how effective these new products can be. For the people that have a lot of Roundup drift, you can make the drift damage disappear by putting on primarily copper. We'll have another slide here in a little bit. Now, tar spot showed up for the first time probably 3 or 4 years ago. Story in Successful Farming, our farm journal, was quoting Missy Bauer, who runs with a fellow in Illinois for that farm magazine, and they were reporting 60 to 100 bushel acre yield loss from tar spot. Darcy Terlenko is running the 8-state tar spot project for testing for fungicide use, and they were using basically scare tactics, in my opinion, and asked her what her tissue test results showed. And she asked, why would I take tissue test? Well, I put Carol to work with myself. I showed those pictures of those leaves to the Iowa State pathologist, and I said at a field day, I think we can help you on tar spot. She said, I don't think so. It's a tough disease. It's a different sort of disease. And I told her I wanted to do some work in the greenhouse that next year, or next summer, or winter. And she says it's a tough disease to work with because it's the only fungus that you can't scrape the lesions off the leaf. And it's the only one where the spores and the lesions do not germinate. So she called me 2 weeks later and asked for if I could get some powder to her. I asked, when do you need them by? I'll get them to you. And I said in return, I want 10 leaves from each of the 3 sections of the plot. Well, I got her the products, she got me the leaves. The next Tuesday, they were in a paper bag, stored in a refrigerator. I took them home, I stopped and bought another paper punch, and Carol and I sat up that night and used a paper punch to punch out the areas of leaves with the greatest concentration of the spots. We sent them to a friend of Don's that has an X-ray diffraction scanner. What we found out is that when the zinc levels are half what they should be, and the copper levels are less than half what they should be, and the sulfur levels are extremely low, Don analyzed the whole thing, and I've got the sheet in print form, but he says he saw those same types of levels in 3 other crops: oil palm, bananas, and corn that has Gauss's wilt. And he said, I know exactly what's going on. He said when you got those minerals lacking, the phylochora fungus, which is a weak fungus, cannot process the sugar in the form it's coming into the plant, so it spits the excess manganese out onto the surface of the leaf, and it's in the form of a manganese oxide. So I would call it, it's more like welding slag. So, Alan, can you wipe welding slag off a piece of metal with your hand?
No.
You gotta grind it off. That's why she couldn't scrape the lesions off the leaf. And why the spores don't sporulate in those lesions, they're sealed inside a manganese oxide tube. So, put that knowledge to use, and we put together a management plan to handle tar spot. And what we're saying is we want to take tissue tests, probably at V6, V7, and then again V12 or V14 to make sure you're meeting the critical levels that are needed to keep the plant healthy. Now, as far as what we saw with results, I used that knowledge and recipe last 2 years on 1,250 acres of corn each year in near Monroe, Wisconsin, which is a hot spot for a tar spot. Evan, were you satisfied with the corn yields last 2 years?
Yes, sir. Some of the best corn yields I've had.
In a bad area. You want to come up and,
Well, I can just tell you that if I talk loud enough here, I followed Bob's program. Not that I didn't believe him, but I put on some fungicide as well. And more of a farmer example, a creek splits the farm, low ground on both sides, and saw no yield difference in where I followed Bob's program. The bio-improve, what else was I going to say? And then talking to some farmer friends who actually tell the truth on their yields that were applying fungicides a couple of times, my yields were right there with theirs.
yeah, we didn't use any hard fungicides. I looked at quite a few tar spot fields in northeast Iowa and southeast Minnesota. Most cases, I had to tell the farmer, your corn's dead, but it wasn't tar spot that killed it. It was Gauss's wilt that got it. So if all you do is put on the minerals, and Gauss's wilt still kills it, and if we're dealing with $6 corn again next year, and a full rate of bio-improve is going to cost you $14, would you spend $14 to get 60 or 100 extra bushel? That's what we're seeing. But we're eliminating the nutrient deficiency, and we're keeping the fungus at bay. So I got the critical levels from Ray Ward at Ward Labs. I talked to Dave Sassville, got his levels, and talked to Jason Schley and got his levels. So we got a chart, a grid put together. To continue going, how exactly do herbicides kill plants? Well, what they do is they create a mineral deficiency that allows the microbes to come in and cause root rots. I dug this up. What minerals are chelated by different herbicide families? You ever seen this chart? I don't remember getting it, but I had it. But each of them, when you read about a new herbicide coming out, they'll say that it operates by shutting down an enzyme system. what happens when you shut down an enzyme system is you pull away one of the crucial metals for its metabolism. And a common one, say sulfonylureas, chelates phosphorus. If you have a bleaching herbicide, typically, in order to make chlorophyll, you have to use magnesium. So the bleaching herbicides chelate magnesium. And so on. Now, glyphosate is a broad-spectrum chelator, so it chelates zinc, manganese, cobalt, copper, boron, and potassium. That's why they say it's a broad-spectrum chelator. A couple of them, they haven't declared what they were for sure, so I still have a few question marks in there.
Did Don tell us that Liberty or glufosinate worked by chelating cobalt?
Yeah, that's one of them. Yeah, that's one of them. We're waiting for a couple of the gaps to be filled there. And I think I know where this is coming from. Big farm in Guatemala. Okay, so I said, well, I've got Martha here is we finally have a product now that we can use to break Roundup down in the soil. And say, Evan's got a farmer in neighbor close by. I think they're farming 30,000+ acres of corn, and supposedly they spray 2 or 3 times with a fungicide and just couldn't control it. Because southeast Iowa last year, the top rated fungicide did not work because it's not going to work. It's not a fungal disease, it's a deficiency problem. So what we used then, we went to the company called Mesalosate because we ended up finding out that one of the spray planes that we tried to load the manganese on had to come back with a tank full, and I don't want to be responsible for creating a young widow. So they had a better chemist who happens to be Don Huber's first cousin's nephew. And anyhow, they've got a full lineup of products. As far as quality, my doctor who told me I should start taking magnesium, I went to buy magnesium, and guess who made 50% of the top rated chelated minerals for human health? Same company. So how many people use foliar feeding? No, you're top growers then. Did you ever find a whole pile load of information on how valuable foliar feeding was? I couldn't either. I looked for 20 years. I kept getting referred to Michigan State. Never could find anything. I kept looking for a guy named Morris Fatosh, who was the principal research scientist up there. Ended up being a guy named Harold Bradford Tookey. Now, I've got a sheet. I found an 8-page testimony he made to the U.S. Congress on the value of foliar feeding. He was in the horticulture department, and he went out and he obtained radioactively labeled fertilizer from the Atomic Energy Commission, and he sprayed it on plants. Some were woody plants, some were vegetable plants, others were row crop, and he applied it, and then after certain periods of time, at divide 24, 1 hour, 2 hours, 3, 4, 6, 8, 12, 24 hours, he went out and cut off plant parts and laid them on an X-ray scanner and measured how fast they moved and in what direction. And then he created a chart that said, here's the efficiency ratios of these minerals put on the soil versus foliar applied. Phosphorus is 20-to-1. Potassium, nitrogen, 10-to-1, copper, 35 to 38-to-1. Now, I really have my suspicions as why this information got withheld from the public. Would you?
And chemical companies didn't want that, but fertilizer companies.
Well, I was at a conference out in Indiana, and Elaine Ingham had been there a couple years earlier. And someone asked her if she could provide them with a list. She said she'd have it there the next year. She came back the next year and they reminded her of the promise. And she says, I don't remember making that promise. If I did, they'd kill me. So Anyhow, when we do spray foliar fertilizer on, you want to spray at the right time, plant growth wise. You want to spray when the air temps are less than 85 degrees. You want to drop the water pH, and then you want to prevent wash off. And to do the preventing wash off, we use a product called Argosy, and that is a medical grade polymer. When they insert a heart stent, they have to slow release anti-rejection drugs, and this is the polymer they use to do it with. Yes?
Does each mineral have a different pH that the water needs to be at?
You typically want to be a half point below what the leaf pH is. So the average recommended pH is probably about a 5.5 pH for the water. And George talked, he's probably got one of the most effective ones that works, that uses the least volume of product. And then also, it tends to hold steady at 5.5. So that would probably be the one I'd recommend. Indicate 5 had done really well, but all of a sudden we found out it wasn't perfect because it's a phosphite-based product. So last thing I thought I'd put up there, because it looks like we're going to have to face dry conditions the next few years. But I do like a product called calcium silicate or mainstay. A couple reasons. It makes the stalks much stronger. It will increase water use efficiency by 36%. And in Brazil, South America, they use it, and Asia, they use it as a fungicide because it will make the cuticle structurally stronger, and your infection pegs cannot infect nearly as easily. Yield-wise, Dave at Guthrie Center was seeing 32 bushel 1 year. Guys in South Dakota were seeing 24 to 40 bushel with it for a cost of $7.50 an acre. And how it does it, this is from a medical doctor, is it creates a more intact vascular system. So it's just kind of like if you're trying to water something and you got a water hose that's got 8 holes in it. This plugs the holes. And research-wise, we worked with a guy named Dan Scow. Scow learned from Reams. Reams was a mathematician on the Manhattan Project. Every product that Reams created, he had some silica in it because it was very beneficial for a number of reasons. When you spray it, you'll create a thicker leaf that you can actually measure the difference with a micrometer. But a thicker, deeper layer of cells forms that hold the photons, so you catch more chloroplast, which capture energy from the sun. So we like what we see. Also, we use a product called MycoGold, which is mycorrhizal fungi that comes in a dry packet that contains both Bavaria bassiana and mycelial fungal strands, so you grow fungal strands, roots out, and extend the root system maybe 3x to 5x, make it a lot more drought tolerant. Hybrid-wise, I like the B37 hybrids, which more the Pioneer than Beck's hybrids, and stay away from the B14s because they're have a shallower root system, wide but shallow, and then also they're more susceptible to goss as well. As far as, I like raising peaches and fruit crops. This is a couple of years ago, we went out on a Saturday morning and picked 5 big boxes of peaches that range from baseball size to softball size with the bricks of 15 to 16. But since then, we haven't had a good crop because it gets below 21 below the zero, the things freeze. So with that, any questions?
Are you still using some products from Montgomery? Are you using products that makes it more water use efficiency?
Yeah, one product I wanted to mention. We're using one called Bio Ensure, and what it is a fungal product from Rusty Rodriguez. And what it does is he found it along with his grad students when they were out in Yellowstone, and they noticed, my wife and I were out there in 2020, And when you go through Yellowstone, there's a lot of geyser activity, and they've got them fenced off, and there's warning signs there that say, do not walk on what looks like a solid surface, because if you fall through, the water's 160 degrees, we cannot rescue you, you will stew to death. Well, they saw plants living where that water was running, and they thought, how can those plants grow? And they found a trichoderma fungus that was living in the air cells in the stem and the roots, and it allowed the plants to tolerate temps up to 164 degrees. So, guy that was here yesterday put some on his corn in the middle of the drought up by Laverne, Iowa, and he, no lie, he told me the yield checks were showing he was 80 bushel better in those areas than he was with than without. That was amazing. Typically 20, 30 to 40. Guy in Nebraska a couple years ago, he said easily 25 bushel. So cost-wise, if you put it on the seed, it's about $4 an acre. In season, it's about $8 an acre, but we like what we see. But it's another trichoderma fungus, which is the same as the Saber-X trichoderma. They all come out of, typically, from Gary Harmon's program at Cornell University. Any other questions? Yes?
Have you noticed some of the rootworm traits aren't performing like they should?
Yeah, most definitely. See, people like to say they're infallible and all-knowing and all-seeing. And anyhow, so often they don't pay attention to some problems that, or I should say traits that set insects apart. In the case of rootworm, Marlon Rice was the entomologist at Iowa State, and Kevin Steffi, who was a senior when I was a sophomore, or a grad student when I was a sophomore, as a sophomore at Iowa State, was in the same class I was. And he ended up becoming the entomology extension researcher at University of Illinois. And he went out and he took 5 varieties from DeKalb, and he planted them, and he went out and he analyzed them for protein, the delta protein concentration in the plant. And he found out that one of the hybrids never had enough toxin in it to kill anything. 2 had just barely enough to kill the larva at V4, but not V9, and it wasn't effective at all, V9. And one was quite effective at V4, but so-so at V9. And I asked him at a meeting, does that mean that corn companies should list the concentration of the delta endotoxin by variety? And he said they should, but they never will. Now, Bruce Tabashnik did a research project with the Chinese entomologist, and they're looking at earworm because you have 6 generations per year in an earworm population, and you can crank through 37 generations in a little over 6 years. They did that. In the end, they came to the conclusion, usually you got a dominant gene or recessive gene. And you can have, if you cross male and female, dominant, you get a dominant one. If you cross dominant, recessive, you still get a dominant one. If you cross recessive and recessive, you get a susceptible. And they found out there was another category that they didn't know existed. It's called a non-recessive gene. And that let the bugs slip by, and that's why we're seeing it fail to a great degree. Now, I'm trying to resurrect the program, but Marv and I used to sell a whole semi-load of watermelon juice every year. And what it was is the original ancestor to our cantaloupe, pumpkin, squash, was a small, spiny melon that was very bitter. And the whole family is called cucurbitaceae, or cucurbit family. And Iowa State is a plant introduction center for them, and they come from the Mediterranean area. So what, they found a genetic throwback melon at the Beltsville, Maryland Research Station by the entomologist, and two brothers from Eustis, Florida would take those melons, squeeze them out, gather the juice, and then they turn it into an emulsifiable concentrate. And we would spray that on the cornfield at like 8 ounces per acre, and we spray alternate strips through a field. Nowadays, I would spray 4 rows every 40 rows, but we would do that right when the first females were puffing up, and when you squeeze them, the eggs were individualized. We knew they were going to be hatched or laid shortly, but we'd spray it and put 1/10 of a rated pen cap with it, which cost $2.50 an acre. We'd go into a field and spray alternate strips. So the guy, you sprayed a lot of it, Ralph. And anyhow, we pretty much wiped out all the egg layers and all the egg laying for roughly 25 days per application at a cost of about $7 an acre. But if you say we only sprayed half the acres, it was $3.50 an acre. Now with drones, if we can find a substitute or have an artificial scent made, we could go out there with I would use Stewart, that's what we tried. Very good results, but we could spray 1/10 of it because with a drone now, you could spray 4 rows, leave 36, spray 4 rows with probably 8 ounces of the attractant and 1.5 ounces of Stewart on 1/10 of the acres. Could be incredibly cheap, so that's a project I'm trying to work on. I've got schematics of the cucurbitaceans molecule, yeah.
Have you done any work with Lithobit?
Oh, Kent Fredericks is probably the most knowledgeable about that. It's a calcium that has a negative charge on it, coming out of Germany. Is there some place in the U.S. that's making that now?
Not that I know of.
Trust the Germans to find out how to violate Mother Nature, because calcium is supposed to be a positive charge all the time. But I do know Mike McNeil was here earlier. Him and Kent worked together. They raised aronia berries. Most people's aronia berries taste like sour cardboard. His tastes like cherries, and he's in the medicinal market with it, and works quite well. Mark?
What's your thoughts on seed treatments and seed treatments, and let's get into the pivot talk, pivot bio talk.
Oh,
I know that's open to a can of worms.
yeah. I had the guy invited that is the expert on it, but he called me and he said he had to bow out for a specific reason, and I wasn't going to press him on it. I told him, do what you have to do. Mark's asking, how about Pivot Bio versus Invita? And Barb and I were in Tony Hagen's research lab out in Tees, South Dakota. Tony's one of the smartest, most innovative scientists I've run across anywhere in any lab. And asked him the question, which one is best? He says, they all have their strength. Pivot Bio lives in the root, and it fixes a lot of nitrogen in the first 20 or 30 days, but then it also starts tapering off. Invita tends to take a while to build up in population, start pumping out nitrogen, and then you got the azotobacters and azospirillums, which are in the USDA collection, and they all have their strength, they all have their weakness. And I asked, what do you expect to happen? He said, if I was in charge, I would have all but one of the companies go broke. I asked, why is that? He said, because the survivor would put all those different microbes in one batch, and then you'd have the greatest stretch duration, production during the season, and it would be all the way through the stalk, and production maybe long enough to make it worthwhile. So really what Pivot Bio is doing is talking to the top fertility guys. They said normally people over apply by about 20% on corn. That's what the research shows. So they're kind of trying to claim that 20% as being how much their product contributes, but really they're not. So at the high price tag right now, I'm always real leery about companies that are funded with Silicon Valley money.
Would you rather treat your seeds with a biological proven seed treatment or stay with the fungicides and insecticides?
Spray tech is doing enough right now with minerals and amino acids. I would sooner put on a biological and a mineral-based seed treatment. And I think in most cases, we'll see the value of fungicides fade away. And when they have the meeting between the American Medical Association versus USDA, the handwriting is on the wall. Now, I mentioned earlier, in South America and Paraguay, all the hard fungicides are mixed with a spray tech mineral, amino acid, phosphite to have any efficacy at all. And I've been told by a real good source, you're going to start seeing the big fungicide companies put the minerals in their products to have any efficacy in the future. So my take on that, I'm going to do sampling, and I'm ready to send the samples to a lab and find out if they are putting them in there. Because the fungicide companies are not going to want to give up any marketplace. So they're going to try to stay alive and hold their market share. So it's going to be interesting.
You think we can develop or find seed growers that are going to grow a viable enough seed with enough vigor that we can, I mean, not so much bypass the biological, but help aid in that?
I'm really disappointed that there's only one seed company that actually fertilizes the seed fields for optimum plant health. That's Gilbert Hostetler down there. Syngenta stayed away from killing their seed with salt in the fall of the year because they found out it cut total seed volume weight by 36%. And a lot of times it's the oils and the minerals get put in the seed last, and those are crucial for good emergence and early plant health. Kevin Kimberly irks a lot of the seed companies because he has his farmers send in a lot of samples from their biggest lot seed for private germination test, cold test. And seed companies don't like it when someone brings 500 bags of seed back because they say it's low volume. But I tell guys, if you're going to plant seed early, choose the larger seeded varieties because they got the most vigor. But when they kill the seed with salt water, and I learned yesterday, a lot of the Monsanto seed fields are being sprayed twice with glyphosate. That's not going to create a healthy baby. This country, when a woman is pregnant, she is told, take your vitamins for a healthy development and a good child, healthy child. Seed companies are doing kind of the opposite. Like I said, I worked with them for 20 years. They're not too smart oftentimes. They can advertise well.
Is it worth trying to raise our own seed at that point, find some older off-patent varieties?
There was one variety that Don worked with, a plant breeder at Blacksburg, Virginia, and every plant would put on 6 or 8 years. So instead of planting 30,000 per acre, they plant 18 or 20 and they had a bigger stock. But it's kind of like, you never read many stories about Jimmy Fredericks down in rural Nebraska. He's raising 130 to 60 bushel beans with populations of 50 to 70,000. And he says, give me good biology. I don't care about tissue, soil test levels for minerals. Make the minerals that are there available. Any other questions? If not, we'll give Mark the floor for a little bit. So thanks for your attention.
Oh, gee.